Welcome to Office Hours today. We're incredibly excited that you are here with us today. Before we get started, I do have a few announcements I would like to make and go through before I introduce our speakers today. My name is Sam Stewart. I work at the Data and Research Center for the All of Us Research program. And so we are going to have a wonderful office hours focused on genomics. So hopefully everyone can hear the audio okay. At any time, if you have questions throughout the presentation, feel free to drop them in the chat. I'm going to go over these announcements and then I'm going to turn it over to our speaker. So the first announcement I want to announce today is that we have a new featured workspace that's currently available on the Researcher Workbench. Dr. Alex Beck and the research support team have created this featured workspace to highlight how a GWAS can be created in the All of Us research program platform, specifically the Researcher Workbench. It really aimed to answer the questions of run, cost, and time. And so how to create an efficient and scalable GWAS study. I also want to shout out Jennifer and Henry, who are part of our support team that helped make this effort possible. So if you navigate to the featured workspace tabs under the demonstration project section of the featured workspaces, you should be able to find this workspace. I also want to announce another opportunity for you to be involved as well. So there are a growing number of large cohort available in biomedical research, including the All of Us dataset. And these cohorts are valuable individually and can be even more valuable if we combine them through cross-cohort analysis. So there are some barriers to cross-cohort analysis, such as data access, data harmonization, and different data analysis tools. So leaders of the All of Us research program, Genomics England, and UK Biobank are gathering a list of potential cross-cohort analysis projects to better understand the barriers to such projects and solutions. So if you have some insight into this and you want to give input, we would be interested from hearing from you. So, and the cohorts that you are using and the way that you're using cross-cohort analysis. So if you follow the link on your screen, this gives you to a short survey that you can answer a few questions and share your thoughts. So I will put that link in the chat in just a moment if you are interested in that. And the last announcement I wanted to make is if you are interested in learning more about the All of Us Research program and staying up to date, please subscribe to the monthly All of Us Researcher Roundup newsletter. You can scan the QR code on your screen and learn about funding opportunities, researcher testimonial videos, and other news around the All of Us Research program that is specifically catered for the researcher side of the program. So now I'm going to introduce our speaker today. Jennifer Zhang is a member of the research support team in the All of Us Data and Research Center here at Vanderbilt University Medical Center. She is a wonderful colleague, and we're so excited for her to present on genomics today. Her work mainly focuses on helping researchers utilizing the genomic data and tools within the Researcher Workbench to facilitate their research. So I am going to stop sharing my screen and pass the mic to her. I do kindly ask that if you could keep your microphones off or stay muted to the end of the presentation, or if you have questions, you can put them in the chat. So Jennifer, I will leave it to you.
Okay, thank you, Sam. Let me share my screen. So even though it is called the Genomics new user orientation, so users who already have had previous experience with genomic data will also benefit from today's presentation, since we just released a new version of the genomic dataset in the Research Workbench. So after today's presentation, you should be familiar with the genomic data and the tools in the Research Workbench, and you will be able to start a simple genomics project, know how to access the genomic data in different ways according to your research goals. And you will also be able to utilize the resources in the Research Workbench and User Support Hub to facilitate your research. Okay, I will first give an overview of the genomic data and tools in the Research Workbench. So some of my colleagues have already given the overview of the new genomic data set during the previous office hours, if you have attended that. So today's presentation will a little bit different. It will focus on connecting the data tools and supporting materials. So after the overview, we will jump into a live demo on the Research Workbench. Hopefully, we will have some time to cover some of the frequently asked questions in the end. Okay, let's get into the genomic data. So on the top level, based on the sequencing and the genotyping technology, the new data set can be divided into 3 categories. The short-read whole genome sequencing data, long-read whole genome sequencing data, and microarray genotyping data. In the new data set, also known as V7 data set, more than 245,000 participants have short-read whole genome sequencing data. Comparing with V6 dataset, 248 participants withdrew, and we added more than 147,000 participants into the new dataset. We provide raw data, short variants, and structural variants for the short-read whole genome sequencing data. Since the structural variants is a new variant data type, compared with the V6 dataset, so we only provide structural variants for around 11,000 participants. And the long-read whole genome sequencing data is also a new data type compared with the V6 dataset. So we only provide around 1,000 participants for the long-read whole genome sequencing data. Then we also provide raw data, short variants, and structural variants. And there are around around 313 participants have microarray genotyping data. Compared with V6 dataset, 416 participants withdrew, and we added more than 148 participants into the new dataset. We also provide raw data and short variants for the arrays. So for those who have long-read whole genome sequencing data, also have the short-read whole genome sequencing data. And those who have the short-read whole genome sequencing data also have the arrays. Their relationships like this. If you have attended the general news orientation, you may know that besides the genomic data, we also have other non-genomic data type in the research workbench. So these 2 papers show you the participants who have genomic data, other data types. So many of the genomics researchers are also interested in electronic health records data. So we highlight here that about 84% of the participants who have short-read whole genome sequencing data also have the electronic health records data, and around 82% of the participants who have the arrays, the microarray genotyping data, also have the electronic health records data. Okay, we just learned some information about the participants, the genomics participants. Then we get into the genomic data, the data types. So for the short-read whole genome sequencing data, we provide single sample params as raw data, and we have support article in the user support hub and a tutorial notebook to show you how to analyze the params using the interactive genomics viewer in the research workbench. And we provide short variants in the format of a Hale variant dataset with all the variants in it. And since this is a new file format compared with V6 dataset, so we have a tutorial notebook as well as a supporting article to explain what is a variant dataset and what is inside it, and how to work with the variant dataset. And so since it is expensive, relevantly expensive to work with the variant dataset, we also provide some other common formats for the short variants for only limited regions, like that is the variants in the exome regions and in variants, cleaver variants, and the common variants. And we provide the Hill matrix table format. This Hill matrix table have all the variants in the smaller regions, and we provided 2 versions: one with biallelic variants, one with multi-allelic variants. And we also provide VCF files, PLIN files, and the BG files, which are all generated by chromosome for all the small classes. So if you can find more information about the smaller classes in this support article, in the user support. And all the structural variants are stored in a single VCF file that is called merged VCF file. And we provide a support article to show you the quality of the structural variants and also some benchmarks for the structural variants. And besides the raw data, the variant data, we also provide auxiliary data for the short-read whole genome sequence data. Like the journal cost sample QC, the variant annotation table, and the relatedness, genetic predicted ancestry. You can find all this information in our genomic data quality report about how we generated those auxiliary data. And if you want to know more information about the variant annotation table, we also have a support article to explain all the fields in that table. So, and I highly recommend you to read through the section short read whole genome sequencing data in the tutorial notebook. Get started with whole genome sequencing data. It will show you how to access those different files and what is inside in those files. So that is all about the short read whole genome sequencing data. Let's get into the long read whole genome sequencing data. We provide the single sample raw reads in two formats, the GFAs and the FAST files. The GFA is like graphical fragment assembly. And so we also provide the BAM files. That is the file after mapping the raw reads with the reference genome. And we map the raw reads with two different reference genomes, that is T2T and human genome 38. So which means that all downstream files, like all the variant files, we have two versions for both the reference genome. So we provide the short variants in the format of VCFs and the Hill matrix table. VCFs have the single samples and joint core VCFs, and also we provide phase and unphased VCFs. The Hill matrix table is a merged matrix table with all the variants, in the long read whole genome sequencing data, and limited because of the limitation of the Hill matrix table. We only provide the unphased genotype in this matrix table, and we provide structural variants for the long read whole genome sequencing data called by two different tools. The structural variants are stored in single sample VCFs. Besides the raw data, the variant data, we also provide some auxiliary data to show you some features during the mapping process. Okay, that's so. If you want to know more about the long read whole genome sequencing data, we have a tutorial notebook in the section long read whole genome sequencing data to show you how to find those files and what's inside in those files. And then we also have the featured workspace to show you how to work with part of the long read whole genome sequencing, for the long read whole genome sequencing data. Okay, let's get into the last data type, the microarray genotyping data. We provide a single sample IDAs for the arrays, and we also provide short variants in the format of Hill matrix table and PLIN files. These are all merged files. And we have a tutorial notebook to show you how to work with those PLIN files using PLIN as a tool. And so that's all about the genomic data. And if you want to know more details about what are the data type, what are the file types, and where to find those files, and how to work with those files, so I would highly recommend you to read through these two supporting articles, how the off-us genomic data are organized, and the control CDR directory. The second one has all the path to those files. And here the Researcher Workspace work with all of us genomic data, have some tutorial notebooks. I would highly recommend you to read through this Researcher Workspace if you want to start a genomics project in the Researcher Workbench. Okay, so that's all about the data. Let's get into the tools. Since the researchers will work on the Jupyter notebook to perform their analysis, and so we have pre-installed most of the common R and Python packages in the Researcher Workbench. So some of the common tools for genome analysis are also pre-installed, like HAIL, BioPython, BioManager, Genesis, and GWAS tools. And also, we have pre-installed some common command line tools like Link, SEM tools, VCF tools, BCF tools, and JTK. And we have direct access for you to get into command line, get into a terminal, or also known as a command line interface. If you want to know the tools you want to use, if they are pre-installed in the Researcher Workbench, you can go to this support article and to see if they are pre-installed. If not, we also have a support article to show you how to install the tools in the Researcher Workbench for you to use. So, because the genomic data set is very large, so we provide special computational environment for you, for the researchers to work on the genomic data. Now we have a Spark cluster with Google's Dataproc tools, and we have a video tutorial to explain different cloud computing environments about this. And we provide the support for the workflow support, like a DSUB, Nextflow, Cromwell, and we have Researcher Workspaces and supporting articles to explain those workflows and how to use those workflows to start your analysis in the Researcher Workbench. So if you are not familiar with workflows or you don't want to use workflows, we also provide a single notebook to show you how to run your analysis in the background, so you don't have to keep your notebook active all the time. Okay, so that is a very, very simple overview of the data and the tools in the Researcher Workbench and their corresponding supporting articles and tutorial notebooks in the Researcher Workbench. So then I will jump into a live demo about the genomic data and tools. First, I will get into the genomic data browser. If you have attended the general new user orientation, you may know we have 3 tiers of the data of us platform. We have the public tier, the registered tier, and the controlled tier. So the data browser, it is a public tier. Let me just jump into it. I will show you from scratch to how to use the data browser. So normally I will just search data browser all of us. And then the first one will be the data browser. Get into it. So I'm not using my Researcher Workbench account, so I can still have access to this. And then we scroll down here, have a genomics part. You will see that it has different part of the genomic data. You may notice the numbers are a little bit different from what I have shown in the PowerPoint, since the numbers are rounded to 20 to comply with the of us data and statistics dissemination policy. And even though these, like the long read whole genome sequencing data and structural variants and the arrays are listed here, they are not available to view in the genomic data browser. So you can only view the short variants for the short read whole genome sequencing data. I click on this, and you can search, as it says here, you can search by the gene zombo, search by the variant ID, by RS number, RSID, or genomic region. For demonstration, so most of you are interested in RSID, so probably I will only search for the RSID, then it will show the result of the variant you just searched for. It gives you the variant ID, the gene zombo, the consequences, and the protein change, and like the clinical significance, and some aggregated results for this variant. And so the results in this table are all queried from the variant annotation table I just mentioned in the presentation. And unfortunately, you can't export the results you queried here to your local computer or to the research workbench currently. We are working on this as a new feature. It's on our roadmap. Okay, so you can click here to get more information about this variant. These are the same in the table. And you can get the prevalence of this variant in the subpopulations in the OVAS cohort. Okay, so let me see what else I want to show. Okay, here we have a demographics for all the cohort, right? For all the data types, like the long read, the short read, the structural variants, and the genotype, the arrays. You click on here, you will know what is their race, ethnicity, and the sex assigned at birth, and the current age, like this. So I think that's all I want to show in the genomic data browser. Let me see what is next. So next, I will go to the research workbench and to show you access the genomic data using different ways. Okay, I sign into my research workbench account. Okay, I already created a workspace for this. Click on it. So,
hey, Jennifer,
yes.
I think we can't see your current screen. Are you using a different screen for your Researcher Workbench account?
Oh, okay. So let me stop sharing. Let me share again. Oh, screen. Thank you, Sam.
Of course. Yes, we can see it now. Thank you.
Awesome. So we from scratch. Okay. So I log into my Research Workbench account and I sign in. Then you will see the workspaces you created. So I create a workspace for today's presentation. So as I mentioned, there are two ways to access the genomic data. The first one is to use the Cohort and Dataset builder. That we have a supporting article for this to show you how to use this tool to query some genomic data. Let me show you in the Research Workbench. So first, I will create a cohort. And I won't get into many details because we don't have that much time to cover those details. It worth a whole office hour to cover it. So I will just select short read whole genome sequencing data. I will see that there are 245,000 participants here. And you can see the race, some demographics about this cohort. And I want to name it to a smaller cohort. And I limited it to 3. And I finish and review. And I save this criteria. Let me see how many left. And we still have 145, almost 100,000. Okay, so I want to add one more. I will read to age. Following. Okay. select, and finish and review. So save criteria. Let me see. Okay, awesome. We only have one participant left. So, and then we create a cohort and we say one srWGS short genome sequencing lrWGS text. Save. Okay, then we have the cohort. I will create a data set so that this is the one I just created. I've already created one to practice before the presentation. So then we scroll down to, you will see here is short read whole genome sequencing data. So using the cohort and the dataset builder, you can only query the short read whole genome sequencing data. And the long read, the structural variants, and the arrays, they are not available through the cohort and the dataset builder. And then select the VCFs. And also, I want to mention that the genomic data you queried using the cohort and dataset builder are a list of VCFs. Okay, so I create a dataset. And name it as one sample short read whole genome sequencing and save. Then I will analyze it. So it will pop up a window to show you that how much it will cost to extract the genomic data. And so normally we recommend it. So if you click on this support article, it will show you that we recommend you to limit your cohort to under 5,000 if you want to use this tool to extract the genomic data. That is so we recommended this based on the time and the cost, since we have a different way to access the genomic data. Okay, so extract and continue. Then it will ask you to create a notebook, or you can add to an existing notebook. So create a notebook, and simple short read list. And you can select your language, and select Python or R, and then you can select the tool you want to use, and select table. Actually, you can preview the code. So the code will provide you, this is to check if the extraction process is done, and it will provide you some code to merge the extracted VCFs in your bucket. Okay, so you can also select link, and it will provide the code to merge the VCFs. It might be the same as VCF. Okay, I will just select Hill, since I will introduce Hill from the other way. Export. After I export, it will create this notebook, and at the same time, you see there is a genome extraction icon here. I click on this. It will show you the status of this extraction. So once the name of this extraction and the status of this extraction and the data when it is started. So once this extraction is done, it will give you a green check in this status column, and it will show you the size of the genomic data extracted and how much time it took to extract the genomic data. And then you can also view the path of the genomic data you extracted. So for this demonstration, since I've already done this exact same extraction before, I won't wait until this one is done. I will use a different notebook, which for this extraction, it is basically the same, exactly the same, just the path is different. I added, since this extraction is done, so those genomic data are stored in my workspace bucket, and I should be able to run these cells. So this just create an environment variable for the path of the genomic data you extracted. It should say that like the VCF instruction has completed, you can continue. We can continue to merge the VCFs we extracted. So this cell is just to import the packages we need. So we import HAIL. Please note that HAIL is only available in the HAIL analysis environment. So we have other sessions talking about introduce different analysis environment. So I just select a HAIL analysis environment here for this demonstration. Okay, then I can merge. These all are pre-generated code for you to use to merge the genomic data. So it takes time, so I won't show you too much. So then you can just run this. Okay, oh, it's pretty fast. Yeah, based on my experience, it's takes some time to merge the matrix, the VCFs to a matrix table. And I think better don't wait for it. And then you can just run the code below to see how many variants, how many samples are there in the matrix table. Okay, I will just stop. And okay, then for other, I will jump into the next one to access the genomic data with notebook. Let me see this next one. Yes, so the next one is to access the genomic data within the notebook using the path of those files. Okay. So here we will introduce some concept like the environment variables like GS util tools. Normally I will just normally you only need to remember one environment variable, that is CDR storage power. So you need to only remember this environment variable. Get this. I use this function always to get env to get the content of this environment variable. So this will give me the path where the genomic data are stored. So then I will just use GS util tools. So you need to use the flag view to get access to the genomic data, because the genomic bucket is a repository type bucket. You need to use this flag, and then I will just use ls to list the content in the genomic bucket. You will see that we have the whole genome sequencing data and the microarray data, microarray genotyping data. Then if I want to see what's inside the whole genome sequencing data, I will just use this path. And then we see we have the raw data, we have the long read data, short variant data, short read variant data. And then we go to, so for example, if I want to take a look at what's inside the short read data, I use this. But as we mentioned, for the short read whole genome sequencing data, we have the short variants, that is SNP and indels, and the structural variants. So for example, if I want to see what's inside the seed index, I just use this. It's pretty simple, just use this again and again. And then, for example, I want to see what's inside this box file, and these are the smaller classes I mentioned in the presentation, and this is the VDS. So for example, I want to see the common variants, the ACAF. And as I mentioned, we provide this in different file formats. The Hill matrix table, 2 versions of the Hill matrix table, one with biallelic variants and one with multi-allelic variants. And then we have the pink files, and then we have the VCF files and the Bayesian files. So this band file is just the intervals for all the variants in this smaller class. So for example, I want to take a look at this, probably this matrix table with only biallelic variants. Now I have a matrix table for these small classes. It's the same file format as you see here. I can merge these VCFs into a matrix table. Then I will introduce a tool that is Hill to read this matrix table and start the code because I just imported during this cell. So I will import again and image that import equals page 38. So that is the reference genome we use for the short read whole genome sequencing data. And so we run it. So here we can create a variable matrix table equals the path we just get. And then we can just read it. So matrix table equals which read matrix table. It's pretty fast. Should be fast. Okay, then I will show you some functions. Like previously I didn't run in this notebook. So we can see how many variants and how many samples are there. So we'll see there this number of variants and this number of samples in this matrix table. And so that's all I want to show for accessing the genomic data. Then let's see next. So next I will show you how to install some of the packages in the Researcher Workbench. Let's to install tools. For example, if I want to have a Venn plot, I just showed in the presentation in the Python notebook, I will just write from Venn import. Let's see. I said no module named Venn means that this tool is not pre-installed in the Researcher Workbench. So we want to install it. So for the most of the Python packages, we just use pip install Venn. Very simple. It's collecting a package and installing some prerequisite. It is done, yeah, pretty fast. So sometimes you need to restart the kernel, sometimes you not. I will see if I need to. Okay, just rerun this cell. Okay, it worked. So I don't need to restart my kernel. So that is a very simple example to install some Python packages. The next one will be install the R package. So Probably I can change the code. So it takes some time to change the code. Please don't do this during your real analysis. I just don't want to create a new or a notebook during the demonstration. So the same, if I want to import the package GGB, let's see, it says like no package called GGB. So I will just use install the packages. And GGB. Install the packages. Okay, and then it is collecting all the prerequisite and all the information to install. Pretty fast. Okay, we'll see if this works. Cool, it worked. So that is a simple example to install the R package. Okay, let's change back the kernel. So then I will show you an example to install the command line tool. So for most of the command line tools, you can install from the source code. So if the developers have put the code in GitHub, you can normally get the source code from GitHub, and then install in a Researcher Workbench. I will use back tools as an example here. So normally I will just search for this tool and to see if there is any documentation. This process a little bit complicated, not as straightforward as using the pip install or conda install or R install. So because conda install is not allowed for researchers, and most of the tools will have the documentation to show you how to install those from source code. And let me just install. Search this the better tools install, and there is a documentation of this. And we'll see it is still stable. And I think it should, yes, like here it is, compiling from the source via GitHub. So what I will do is I will just copy all this to the notebook. So normally, so basically you can run the command line, like bash scripts within the notebook, just to add this sign on top of the cell, then you can run this code. But normally I will just go to terminal and run this, because for some of the packages it will ask you to enter yes or no if you want to proceed. So here you click open, and then you click on new terminal, then you're into the terminal. So what I will do is I will just copy those commands here. So this code is just download the file from the GitHub, and then we will, and let me just run one more. So we are downloading the data from GitHub. It's very fast. And then we unzip. Okay, before we unzip, let's see if the file is here. We'll see that. So this file is downloaded. Then we unzip it. Okay, very fast. So you see, this is the directory that is unzipped from this compressed file. And then we cd to this one. Just follow the instructions here. cd to PCF tools, and then we see what's inside this directory. Then so the bin file is empty file. And then we have the data account, and we have the make file. Then that's why, so we can run make directly. And if you read the documentation, it says sometimes you need root access to install this tool. But for researchers in the Research Workbench, you don't have root access, so you can install the tool into the user local bin since we don't have write access to this directory. And what we will do is just we will add argument in this command to ask this tool to be installed in the directory we specified in the command. So make install, and we specify the destination destination because we just install it in home directory. Make install first. So it takes around probably 6 minutes or 8 minutes to install this tool. Then I will check the chat to see if there are any questions I need to cover.
I think we covered them, Jennifer.
Okay, great.
Unless you want to pause here and see if anyone has any specific questions at this point.
Yeah, so just put a question in the chat or unmute if you have any questions.
So if anyone has any questions for Jennifer while this is installing, this would be a great time to kind of pause, unmute, and ask the question, or you can put it in the chat and we can read it aloud for her. I see Patrick just posted in the chat, will CRAN files contain the FASQQ data related to telomeres, or is the data kicked out?
So we don't have those FASTQ data in the CRANs so currently, but if you send a feature request to this program, we will pass this to the program, and then we'll see if we can use this for future versions. See. If we don't have any other questions, I can probably go back to my slides to see if I have anything. So before I get into the frequently asked questions, I want you to call for registration for our hands-on genomics workshop in July. That will happen on July 12th and 13th. This will be an introduction to analyze all of us genomic data, and we will have a hands-on workshop. We'll give an overview of the research bench and show you how to build a cohort, how to extract the genomic data in details, and how to run a GWAS using Hail in the research workbench. So in order to attend this hands-on workshop, we will request that you have a controlled tier access prior to this registration. And I see a question. Oh, Chris has answered that. It's about size. Okay. So, okay, still have some time. Then let me see what I want to cover. So I have some. Yeah.
Can you hear me? This is Patrick. I just asked a question related to telomeres. I didn't specifically was asking if FASTQ data was in the CRAN files.
Yeah.
As I understand, the CRAN files is already aligned data.
Yes.
What I was wondering is if it if the alignment extend into telomeres, because those are highly repetitive elements, and sometimes they're hard to align. And so we're wondering if we could there's been a few papers using a whole genome sequence data to measure telomere length. And we were wondering if we would be able to do that in all of us. So
Yeah, so
the
this
question is really, is there a type of file, of sequencing file that we could use that,
yeah.
so that we can measure telomere length?
Yes, so currently we don't provide a raw arrays. That we only provide the file that after mapping with your reference gene. But
Okay.
it is on our roadmap to provide a raw arrays. I don't have the timeline, but if you submit a request and we can provide this to the program, probably we can have this come out soon.
Okay.
Yeah.
Okay, I think I'll do that now. Thank you.
Yeah, thank you. Let me see.
Hello, my name is Sukhwa.
Yeah.
I'm sorry that I joined in late. I know that today's talk is about this genomic data.
Yes.
I have a very naive question here because I tried to look at the YouTube session about that information, how I save the R file at the end of my workspace. I wanted to save it so that in future I don't have to rerun the code again. I saw this one YouTube in the All of Us research, but they have no sound. There was nobody talking. It was just the big image of it. So I couldn't quite understand how to save the R file once I run all the SQL function that was built in to get the cohort, get the different data frame. So I have a data frame for, let's say, for the patient's questions, age or demographic. I have a data frame related to their medication, a data frame related to their ICD-9, the disease thing. So when I have that different data frame, I wanted to save it so that in future I can just open it rather than have to rerun all the things. So I'm wondering, can you show me on the screen or?
Yeah, sorry for the inconvenience. Can you send a link of that video in the chat so that we can fix that?
Yeah. Okay, sure.
Yeah,
Sounds
thank
good.
you. And then, so for example, I can show you very quick. So for example, you create an R notebook and then you run some code inside it. So for example, create an R notebook text. And then suppose you have run all the code here and you write your code and then you run it and then you can just save it. Then you don't have to do anything else. You save it, okay? And then you, for example, just close it. I did the whole thing. And then you go to your workspace and then you see, go to the notebooks. You will see the notebook I just created is here. It's here. You don't have to do anything specific.
So I thought to rerun the command line to build that cohort in the future.
So you don't have to rerun, you don't have to use the cohort dataset builder to build, to get the SQL code again. They're all in the notebook.
I see, but then, a lot of time when I go through the SQL code, I already analyzed the data, everything, and I just want to open and go to, there was like a video about, I forgot the name of it, I forgot, like bucket, something like that, and I couldn't understand the video.
I see what you mean. So basically you created a data frame for your data set, and then you want to save it.
Yes.
You don't want to run the analysis again.
Yes.
So this one, then you may want to save your file. You save your data set into a file.
Okay.
Into a CSV or TSV file, and then copy it to your workspace bucket. I think Sam can put in the chat about the video or support article about we have.
Yeah, that video is intentionally silent But there's a user support hub article to show you how to save to the workspace bucket. And then there's a couple featured workspaces to show you how to save the data frame. So I will put both of those in the chat.
Oh yeah, that would be great. That would be great. Thank you so much. Thank you.
You're welcome.
Yeah, so let's go back to so this tools is done installing, and we will see, let's see if we can run it. It's all still say genomic confined. Let's see what's inside it. So as we mentioned that we just installed this not in, we installed the tool not in this directory. Okay, let's go to we install into your phone. And we can see there is a user here. Okay, actually, I don't have to do this. It says that the file stored here. Home Jupyter local bin. Okay, I will just cd to this app. And then else. So we can see that all the tools are installed here, and we run that tools. Still can't find. So that is one thing about the Research Workbench. So you need to save this path to your bash RC. Hopefully I hope you can do this and run it. And then refresh your bash RC. So then let's run it again. Great, it worked. So we can see that that is the introduction of this tool. And since we have saved this path to your bash C as an environment variable, so you can actually run this tool in or any other directory. For example, I just go one directory above and then run it, still runs. Or basically, you can even start a new terminal and run it. Yes, it worked. Okay, so just about the time. So that's all I want to show today. So hope it is useful and just pass to say.